Okay, so this week, as you are working on your assignment, you are going to be organizing your research, all the work that you've found up till now, into a PowerPoint template that's provided to you in this week's module. That particular template can be found, right here. When you go to week 10 to 12 module and you go into research organization and analysis PowerPoint, you can see that PowerPoint template right there. So the first thing you want to do is download that. In addition, you also want to download, or not download, but rather open up in separate tabs all your research, those primary and secondary sources that you have found for your topic, and organize them into tabs so that you have easy access to the full text version of those articles. Those full text versions are what you're going to be using to create some of your figures, because obviously you didn't do any data yourself, but rather you were using published research to collect information and data for your hypothesis that you've created on your topic. So that's what we're going to be working with. I've also kind of done a little search in Google to get some images to kind of give me overall general kind of images that I can use in the introduction or other places in the poster where I might need to give some information or some additional kind of cool links to add to it. So I kind of organized all my stuff according to that, and now I'm ready to go ahead and start working on this template. Okay, so now that I'm ready, I'm going to go ahead and I'm going to open up my slides and I'm going to start working with them. So let's go ahead and start with that first part, which is your poster title, obviously. Your poster title should include your independent variable, your dependent variable, and your overall conclusion after doing all this research about your supplement and how it affects the disease or disorder that you chose. So in my case, I'm just going to make, show you examples using saffron and cancer as my idea. So I'm going to go ahead and write my conclusion for that. So maybe I say something like crocin in saffron has strong anti-cancer activity against, has strong anti-cancer activity. Maybe that's all I need to write. But this has clearly mentioned my independent variable, which is saffron, and then my dependent variable is going to be looking at that anti-cancer activity in the cancer cells or in actual patients. And then, I need to have some names of the group members. So I'm just going to write here some random names. And then this last line, which signifies your department, your lab, and then the institute that you're coming from, is kind of standard for all of you. So that remains the same for everyone. The next thing you will do is have the next two slides are going to focus on kind of setting the stage towards your project goal and hypothesis. So this means that you need to put information very carefully tailored to show us why your natural supplement will, work the way you think it will, and what is it about your disease and disorder that the natural supplement can work on. So, this one is going to be all about saffron. For now, I'm just going to write that. And then, you want to remember that this is for a poster and not for an actual paper. So in a paper, you can be a little bit more wordy and give a little bit more details, and that's fine to set the stage. But here in the poster, you don't really have that time, right? Or the space to do long lengthy sentences or long paragraphs to set the stage and to give the introduction and background about your topic. Instead, you want to rely on very carefully tailored bullet points. Think about it kind of like the thesis statements from your individual paragraphs in a 5-paragraph essay, and just write the basic information about your supplement and then pair it with carefully chosen figures and images and schematics to make it an interesting information tidbit about your topic. So in my case, like I said, that I opened up some kind of background info type things that I could add to jazz up my, so to speak, slide. And for these ones, you can copy these images and put them straight in there. You can also cite them if you need, if you have something from an actual paper. You obviously have to cite it. You have a lot of stock images available in here as well that you can use. And I'll show you how to get to those as well. So you could just kind of, use that. I just want to give some examples of different supplements maybe and what it looks like in real life. So I'm just doing a few. And because I'm specifically focusing in on its cancer potential, I'm not looking for, saffron supplements that are there for depression or mood or anxiety or blood pressure or anything else. I'm specifically trying to find things that are going to be tailored towards what I want the reader to take out of my study, and that is to focus on its anti-cancer activity. So I'm trying to set that stage here, so to speak. This looks like an interesting, oh, look at that. I like that. So maybe I'll take that. This looks from an actual paper maybe. So maybe I'll open the paper itself and then, kind of use that. Maybe that's all I need to do. Who knows? So, something interesting that I can add about my topic to make it talk about that. This shows the quercetin and quercetin, the two main ingredients that you find in saffron to have the highest anti-cancer properties. So yeah, why not? You can also use some cool ideas from here to present your figures a particular way if you wanted. Sometimes those ideas are really good, sometimes they work out and you can use them, sometimes they're not so good and you can just not worry about it. You can also directly pick images from online by going to insert online pictures and then going into a search engine through Bing, for example, in here, and I can search for whatever topic it is that I want to insert images from. Didn't open up correctly. So there we go. So I could say saffron, cancer, see what comes up. Some images come up. Ooh, that's interesting. I can say Creative Commons only, or I can have more detailed. And now when I click on these, it can automatically insert them in my actual slide. So, if I wanted, for example, a flower, I can do that, right? And now have it. So I'm actually going to take that flower and put it over here. And maybe I want to add that to my background or to someplace on the top of my poster. So it's a pretty flower. I can add that here. I can also add the Unf logo in here, right? That's another thing that you may want to add to your actual paper. So I'm gonna write down here that Unf logo, and you can get to them directly through the marketing website as well. You just have to be logged into the UNF network, or I can just take one of these also. I can just copy the images and use them as well. And so I'm going to add one of those right here. That's going to again go on my final poster. So it's good to have it handy while I'm thinking about it. And so, I can write down some tidbits about saffron. You can find some of these in one of your review articles. You can also just look that information up. So I think I had a review article open right here. So I can look at this and add a few information about that, just to show you an example. So here's a sentence that I literally copied straight out, which is not what you want to do. And now I can play around with it, and I can say more things about it. Grown are used as a spice and traditional medicine in many cultures, and then especially present throughout the Mediterranean, or whatever you want to say about this part. And then add a few tidbits about what might be important in there. Has several compounds with known anti-cancer activity, right? And some other things about that. So you just want to have no more than 3 to 5 bullet points about your topic. You don't want to make it too wordy, and you don't want to have too much information there. And then this one is going to be all about cancer. So again, you can have, happens due to, occurs due to dysregulated growth. Can cause, can happen in any living organism, and any can occur in any living cell, organ system, right? You can talk about how it can lead to death if not removed, or if not processed at the right time. Or you can talk about, considering that you're going to be looking at saffron as one of the things, you can talk about what type of things are important there. Cancer cells can migrate and invade other areas of the body if left unchecked, right? Leading to metastatic. So that's just some things that it can do, right? Going back, we could find more information about saffron and what type of things it may help with. So cancer, no, saffron, and cell growth. So yeah, ooh, that has some good things we can add later on. Okay. Yeah, that is the one that I chose. That was a good one. Okay, so here we go. So here, if you look at this, it talks about how crocin and quercetin have been known to have certain effects that are important for cancer, to control cancer in general, right? Okay, so our project goal is going to be exactly what you wrote in your assignment before. So that is going to be your big question that you're trying to answer through your research. So does saffron have anti-cancer activity? And then my hypothesis is going to be saffron will have anti-cancer activity through inhibition of growth and activation of apoptosis, for example. That's one example. This has 2 dependent variables it's talking about. Most of you only had 1, and that's fine. So then you get to your research strategy. The research strategy is essentially akin to you guys having a method section in a primary literature article. Now, obviously, you did not do any of the research associated with your topic, but you did look up articles that did the research, and that's what you're going to be talking about. Now, all of you should have stuff related to clinical studies or some clinical data, some type of research studies, and then some type of mechanistic molecular data. You may not have everything on this list, but you will have something from each type. In my case, I only have maybe a couple of, I'd only have maybe one clinical trial, but the rest of it is all either animal studies or cell culture studies, and that's fine, as long as you have at least cell culture and animal model, so in vitro and in vivo, or human subjects, and then something from here, either animal models or cell culture, or some other combination like that. Everyone should have molecular data, and that means data on DNA or protein. So gene expression analyses, specific genes being upregulated or downregulated, right? They can look at it by doing RT-PCRs or DNA fragmentation analysis to show apoptosis. Any one of those will work. Again, with protein, you can look for protein activity. You can look for protein expression, whether a protein is more expressed or less expressed after treatment with the saffron. Any one of those would work. And then the pathway analysis is something that you may not find on for your topic up front, but it is something that you're going to be making next week in class. So for your reminders for data selection, make sure that you're taking something from each type of your research. You should have some clinical, some cellular, and some molecular data. Clinical can include epidemiological studies, clinical trials, something to do with humans. And then your paper should include, your poster should include at some point some images of either cells or tissue sections or anatomical objects, portions to show response to the supplement or disorder. And then you must have data, molecular data pertaining to proteins or DNA in response to the supplement. So these are required for your final poster, and not having one of these categories will cause you to lose points. Similarly, another thing that you must do in your poster is to create at least one original figure or table where you are taking data from multiple sources, multiple papers, and combining them to make your own graph. Okay? So here is an example of what I might do to do that. One example would be, there's one paper that is looking at the effect of prosin on a particular cancer cell line after radiation therapy, and it is with and without radiation therapy, and it's showing the viability over time or over different concentrations. I have another article right here somewhere. Where is it? There. I have another article that is looking at saffron at different concentrations and looking at another two cell lines, and it's comparing their viability in response to saffron. So this one has a lung cancer cell line, and then I believe a non-malignant normal cell lines. These are normal cells, these are cancer cells, and they're showing that only cancer cells are dying. So that's another paper. Maybe I find a third paper that also shows something similar. So what I can do is I can take a concentration of saffron that is common to both of these studies, and I can use that to create another graph where I just make a bar graph with the three cell lines, the HN5, A549, and the LC line, and then make a graph where I show percent viability in response to a particular concentration of drug for different cell lines, taking into account the multiple papers that I'm looking at and comparing them to each other. Another example would be to take a particular, different doses from different trials and see how much each dose was having an effect in that particular case and comparing them to each other. You could also just make something like this. It's a little bit boring and hard to see, but in a poster session, but still, it is a way that you can make your original table by taking multiple articles with a particular to either different cell lines or different mice studies or different clinical trials, and then writing something about them and their results and the obviously the reference in front of it, and make that your original figure or table as well. So those are some examples of how you can create an original figure or table that is not just taking data that is already presented and copy pasting it, but rather making a graph or a pie chart or a table all by yourself, all on your own. Okay, after that, you have been provided with 3 sets of 2 slides. For each one, you have been given a place to write your title, and then you will create the figure underneath, and then a slide where you will write the caption for that particular figure that you have developed up above. So you will have, like I said, you can have up to 3 slides, and that's what you're going to have these spaces for. So for your slides, what I suggest is that you come up with your figure titles first, and then you go back and populate them with the right type of data. So maybe I say something like, saffron induces anti-cancer effects both in vitro and in vivo. Right? So that means that now I have to put in articles, data that show that it has an effect in vitro, as in cell culture model, and then also show that it has that effect in vivo, in animal model. And then maybe my next slide will have, a different title. We'll say something like saffron and crocin in saffron induces anti-cancer activity or induces apoptosis in cancer cells, right? That's another thing I can put in. Then I need to find articles with data that show that it induces apoptosis specifically in the cancer cells, right? So again, these are some examples of what you can do. So you want to go ahead and make those titles first, make those 3 main conclusions from your data first, and then go back and populate each figure title with appropriate images. So for the first one, it says that, I want to do something about comparing the anti-cancer activity. So I'm going to copy this image. This seems like a good one for in vitro, one of them. I don't want to necessarily show all of them. There are 3 different slides, or there are 3 different graphs. I go back and I look at it and I see what it is that it's showing in those 3 different graphs. If there is something special that I should care about. So this is just over the 3 different time frames. It's 6 hours, 48 hours, and 72 hours. So you know what? I'm only gonna use a 1 time point. Let's see. This one is at 48 hours, so I guess I just want to use B. So I'm just going to crop out panel B and use that because I have another paper that's also showing the effect of, saffron, and it's showing it at 48 hours as well. So maybe that's the one that I want to use. So I'm going to go here, and now I'm going to copy this image, which shows it at 48 hours, and I'm going to put it right next to it. Right? And now I have these 2 images from 2 different papers, and they are both showing in vitro effect of saffron, right? Now, because I am going to be organizing them my own way, I'm not going to have them in the same exact format as they were before. I'll have my own ABCD, so I can change that as I need. But before that is done, I want to make sure that I am actually adding my what is it called, my references. So this one is 2010. Oof, that's a long name, so I will try. And so this one is going to be 2010 And then this can be, this doesn't have to be quite as big, and I'm gonna put that with the article that belongs to, obviously, and with the panel that belongs to that article. I believe this one was it. Yep. Ooh, this has some pictures too. I want those. This is 24. This is 48. Perfect. Let's get them. And perfect. Yeah, those are the two good things from here. So that gives me some images of my cells as well, and that's exactly what I wanted. And these are the cells at 48 hours. Let's see what was different in A, B, and C. They're different concentrations. So A's control B is 500. My other one is at 500 too. So I'm just going to remove the bottom one, because I don't really want to see that one. And then I can put that here. These two are both from the same, thing, so they can go next to each other, maybe, right? And so these can be my A and B, this can be my C, and now I need in vivo, because remember, I told them that I was going to give them in vivo results. Right there. Perfect. So I can go ahead, and I can copy this image, and I can use that as my fourth. And now I can decide whether I want to show both sides, or whether I just want to show one side, or how it is that I would like to show it. So Control D, I'm gonna actually crop them so that I can decide how I want to present it. And there we go. And then now I'm gonna crop this one the other way, right? Because I don't really need all that extra stuff in there. And now I'm gonna make it small so it matches this one. I don't really like the way they wrote this, so Control D. I'm actually gonna do something with that too. Let's make this smaller. Let's make that smaller too. I just didn't like the way that was playing with our stuff. How about that? And then I can select this and group it and make it bigger a little bit if I want to match it up. And then same here, I'm gonna make this bigger to match this up, right? And now those two are from different papers, so I need to be aware of that. And I can maybe put that right there. Look at that. So see, this is how you can organize your data in your own way as well. Another way that we can do that. And now I want to make sure that I am citing all these papers correctly, obviously. So I am going to go ahead and look at this one. And this one was the one that I already did. This one is the mouse xenograph model, right? That we were just looking at. So let's look at this one right here. So this one, again, you can cite, it's Dhar et al., and it is in 2009. So I'm just going to do Control T, and I'm going to write Dhar et al. 2009, right? And then I'm going to write again for the other one, whatever this one was, right? Because this one is not what it says it is. It's also from another paper. So I'm going to go to that paper right here and check this one. And this one is, let's put them, this was 2017. So by doing that, you have now cited all the papers according to which one is coming from where, the data, where it's coming from, and you kind of have a good idea on how you will organize it. you want to make sure that your, images look nice together. They're organized in a way that they look like they belong together. And then you would add your new ABCD according to however you want to present it. And then that's what's going to be important for your caption that you will write. Obviously, I'm not going to sit here and write the caption and everything. I just want to show you a little bit of how we do this. And that's, so this is going to be my A and B. This is going to be my C. And then I'm going to have a D. And that's going to go here. And while I'm doing that, I'm making sure I'm lining them up. I'll make that like this so it can cover it in a second. And I'm going to fill it with white. And now you can't even tell where it was supposed to be. Right? And then once you're happy with your final figure, you select everything, all these ABCDs, the little labels, everything, and you arrange and group it. And now I can move it as a whole and put it wherever I want. And when it's time for you to make your poster, you will just simply copy it and paste it into your final poster. So that's what you're going to do for each one of your figure slides. One of them, remember, will have that original panel that you will create using data from multiple slides. The next thing that you need to do, and this is very important for next week's lab, is that you need to fill up these two tables with the minimum of these 5 nodes. A node is basically just a gene or a protein that is affected by your condition or by your supplement. And by affected, I mean either it is enhanced in its expression or activity, or it is reduced in its expression or activity. And you do that by going into your articles and looking at actual names of genes and proteins that it tells you were affected by your supplements. So this one doesn't really have any, so I'm not going to look at that for now. I believe this one had some that talked about. So it talked about BCL2 and BAX being increased by the quercetin in your samples. It also talked about phosphorylated EGFR being different in the 2 cell lines. So you want to look at those names and you want to show that. So BAX is increased, BCL2 is decreased, I believe, maybe, or increased. We'll have to find out. But definitely BAX is increased. So I'm going to go ahead and write as one of my ones under the supplement, BAX protein is upregulated, right? And then you are going to write the name of the reference that it belongs here, right? So whichever one this is from, you will write that name. So this is TAR A et al. So you'll just write TAR A. And then whatever year that was from, I don't remember. I'm just writing a name for now. Right? So you'll just write something like that. You will go through all your articles and you will do the same for the others, right? So here's another one. This one is looking at another protein that is affected by saffron. So you will just kind of reduces DCLK1 levels in CRC cells. So you will just say DCLK1, right? DCLK1, and this is downregulated. And then the journal, the article that it came from, and that's what you would write there. And this is in colon cancer, and it's looking at its metastatic ability, migration and metastatic potential. So this is saffron could restrict MAC1 dependent cell. So that's what that was. Okay, and this is 2020. This is brand new. And this is in a very good journal. Okay, so that's what you are essentially going to be doing. You're going to go to your different articles and look at that. Now, sometimes your review article may contain a lot of that information as well. I saw some of you had review articles that contained a lot of information on different genes and proteins affected by your supplements. And so that would be, again, another great place to look in those articles. Now, as you go through those, you will be getting data essentially together for what we are going to be doing next week, which is to make your pathway. However, sometimes you'll find a ready-made pathway that you can start with in your papers as well. So in my case, right here, I can see some that are looking at saffron and their potential in different types of diseases. So this is looking at its effect in digestive diseases. But a lot of the things they're talking about are very important in cell cycle too. So I might actually make that and go ahead and put that here as a way for me to get a head start on my work next week on pathway analysis. So saffron is supposed to increase P53, supposed to increase BAX, reduce P21, right? So it's showing you some things that it's going to inhibit and increase. So it's inhibiting CDC25 and cyclin B. Okay, sure, I'll write that. Cyclin B, downregulated. And then, I'm not going to fill it up all the way right now, but I'm just writing some of these things down. So P53, upregulated, right? So you will go ahead and get some ideas on what the things are that are affected by your supplement that can help you generate your work next week. What's this one? Yeah, and just showing more details. So that's how you're going to go through and populate this entire thing. Now, you're going to also look for genes and proteins that are affected by your condition or disorder. Now, cancer has a lot of genes depending on which cancer you're looking at and what it is. So if you have a general condition like that, you'll just kind of look for some common genes that are dysregulated, like P53 is mutated many times in cancer, so you can write that down, or other ones that we've talked about. But outside of that, it's kind of, some of these are going to be not so concrete. These are going to be very specific to your supplement. You want to look for at least one gene or protein that is present in both sides, both as a node, in a condition, as well as in the supplement, because that helps you make a connection between them. After that, the rest of it is kind of self-explanatory. You're going to write some conclusions, again, as bullet points. You're going to write the clinical implications or biological relevance of all these findings that you found, and then your references and acknowledgments. So I hope this helped you figure out how it is that you're going to be filling this PowerPoint out so that you can complete it this week and get a head start on your poster while doing so as well, and also kind of help you organize your thoughts, processes, and help you make your conclusions about your project. Okay,